Okay, so we're going to tackle this second question here. Um, this free response question, and it's talking about cyclin dependent kinase inhibitors. All right, couple things you need to grapple with. All right, so cyclin dependent kinase inhibitors. All right, so if these if cyclin dependent kinases are present and uninhibited, then DNA replication will occur. Okay, so this graph is telling us that this experiment is basically explaining to us that if P16, P18, P21, and P27, they're believed to be kinase inhibitors, if they are present, right, in some percentage, then the cell may not enter into a phase where there's DNA replication, okay, because they're going to inhibit cyclin dependent kinases. So these four proteins, P16, P18, P21, and P27, are present. They're going to inhibit these kinase inhibitors, and then there won't be any DNA replication, right? So that's what they thought, right? All right. And so we read it says scientists use cells called fibroblasts to study factors that cause non-dividing cells in the G0 stage of the cell cycle to re-enter the cell cycle and undergo mitosis. Now remember mitosis, we talk about that. We know it's interphase, prophase, metaphase. Well, interphase and then mitosis, which consists of prophase, metaphase, anaphase, and telophase. All right. And then interphase has that G0, S phase. And so in G1 phase. So we can go back to that and say, well, during this, let's say if we look at what's occurring during the G0 phase, right? And G1. So cells enter the G0 phase from a cell cycle checkpoint in the G1 phase. All right. And it says during the G0 phase, the cell cycle machinery is dismantled and cyclins and cyclin dependent kinases disappear. All right. So without these kinases, the cell is not in a dividing or DNA replicating stage. All right. So we have to look at this data for what it is. Also up here it says the G0 phase or resting phase is the period in the cell cycle in which cells exist in a quiescent state. G0 phase is viewed as either an extended G1 phase where the cell is neither dividing nor preparing to divide. Okay, so it's basically in a resting state. All right. And so if we go back to this thing, right, we realize that non-dividing cells are basically in the G0 stage. All right. So to get them to re-enter, we're trying to figure out, well, what would make them re-enter the cell dividing stage, right? And so we're trying to see if the absence of one or more of these proteins, which will inhibit these cyclin dependent kinases, would cause these cells to re-enter the stage of DNA replication, and that's what we have here. All right. So we're looking at this data. You got to look at it, see what is going on. It's kind of tricky, but plus or minus down here, these P16 to P21 basically is whether this protein is present or not, which is also a kinase inhibitor. So if it's present or not, what is it? What effect does that have on it, right? And the way you need to look at this data is this. Okay, so we can see if we just to say we focus on P16, right? So just to clarify, we focus on P16. We see that without P16, right? Without P16, this protein P16 being present, it's pretty much the same as having, right here all the way to the left, the same percentage of DNA replication when there are no proteins present. So what you want to realize is that P16, the absence of P16 really is having no effect on DNA replication, because it's the same as, not having any proteins. It's the same as here. It's the same effect as, 0% serum, which, remember, these are serum starved fibroblasts. So the absence of P16 is just like saying, okay, this is the normal state when cells are in the G0 phase and there's really a lack of serum, and you're getting about 20% of DNA replication. So, all right, so that's how you look at this one, where P16 is really having no effect on this whole process. Okay, and then you look here. Even you can tell here, if you look at group 7, you'll see that, all right, if we remove all of these other proteins and still have P16 present, then DNA replication still occurs. So these proteins, 18 through 27, have a greater effect on the rate of DNA replication than P16. So when we look at this question, it says, based on the data for the groups of cells cultured with 0.1 serum, which is only groups 3 through 7, it says, identify which of the 4 proteins tested do or does not appear to play a role in blocking cell cycle entry by fibroblasts. So here, this is what the normal cell looks like, when it's starved of serum and it's in the G0 phase, and without P16, it's just basically how it is normally. All right, so it's not really this protein is not playing a role in blocking cell cycle entry by fibroblasts. I think that's how I would interpret that. The response indicates that group 3, in which P16 is blocked, does not appear to play a role in blocking cell cycle re-entry. And then, so we'll move to question B, where it says, based on the data for the groups of cells cultured with 0.1% serum, groups 3 to 7, obviously, identify the groups most likely to include cells undergoing mitosis. Mitosis is cell or DNA replication. So you see all of these groups, because of the percentage as compared to the 10% serum, they have various levels of DNA replication going on, and this is groups 4 through 7. All right, groups 4 through 7. Groups most likely to include cells undergoing mitosis. That's question B. So it says the response indicates that cells undergoing mitosis are most likely to be found in group 7. You could say that, or in groups 4, 5, 6, and 7. So if you just, wanted to look at the one that had the most DNA replication, you could have included that. And this is a point that I'm trying to make. You could say either or, that, these are the scoring guidelines. So they're saying if you just say group 7, you get the point. If you say groups 4 through 7, you still get the point, because there is considerable activity here as far as DNA replication is concerned in groups 4, 5, 6, and 7. All right. And now we try to, I guess this is the analysis or application part here when we do this question. It says neurons. All right. And we should remember that neurons are the cells, typically associated with our nervous system that do not replicate. That's why you can't drink or smoke or suffer damage to your brain or spinal cord, because these cells don't regenerate. They don't replicate again, right? They stay in the G0 phase where they're just doing their job. They're not dividing. So it says brain and spinal cord are in G0 of the cell cycle and generally do not divide in response to an injury. So if something happens, whether that's accident, physical, or chemical damage, these cells are not going to regenerate. So you gotta guard those cells. All right. And so it says the scientists predict that they can use the data from this experiment to help them treat individuals who suffer from spinal cord injuries, and then provide reasoning. So what your job is to provide reasoning to support the scientists' prediction. Well, based on this stuff, the normal thing that happens is these proteins are present, right? And they're looking at 4 of them. So they're trying to figure out which ones are causal or which ones are involved in inhibiting these, DNA replication. And we can see here that, 18, 21, and 27, they're very much involved in that. So what if we could, reintroduce, right, these proteins here, okay, and somehow force the cell to, or get rid of these, get rid of these proteins and force the cell back into the DNA replication cycle, right? Based on this data, we can do that, right? Based on it, well, it's a possibility. I'm just saying that can be done if these inhibitors, these cyclin dependent kinase inhibitors, are not found anymore. If they're blocked, right? If we block the presence of these, then the cell will jump back into DNA replication. So if we can eliminate these things, right? If we can block them in some kind of way, we really can't eliminate them. We can block them in some kind of way, the cell will jump out of G0 and back into the cell cycle and divide. Okay, so that's what we can do based on this data. So provide reasoning to support the scientist's prediction. Let's go to C. And we'll say the response indicated because blocking some of the cyclin-dependent kinase inhibitors, which are 18, 21, 27, made fibroblasts divide, the scientists might also successfully block different cyclin-dependent kinase inhibitors in neurons of the spinal cord to make the neurons re-enter the cell cycle and divide. So it's sort of backwards thinking, like, okay, if we block these inhibitors whose job is to block cyclin-dependent kinases, then we can, make the force neurons to make more neurons, okay, and then basically heal itself, all right, or heal that system. So then it says interactions between cyclins and cyclodependent kinases control the cell cycle. Okay. Explain how the presence or absence of inhibitors of cyclin-dependent kinases might play a role in normal cells becoming cancer cells. Okay, so the presence or absence of inhibitors, right? So we see here if these particular ones, for example, are not present, then these cells replicate, right? So part of the normal cell's process is to stop replication, right? And allow the cell to do its work, right? And so we have these people in here, these proteins in here, they're like guardians. They're saying, all right, we're here to stop DNA replication. Okay. And we realize that uncontrolled DNA replication or uncontrolled cell replication is cancer. You got to think about that. The cell's dividing constantly, that is cancer. So if we extrapolate that a little bit, we say, well, okay, that could be caused by the lack of 18, 21, and 27 from this data. We're looking at P16. P16 doesn't really have an effect. Even if it's present, there's still DNA replication. So this could have implications on or the presence of these proteins, which are cyclin dependent kinase inhibitors, could have an effect on uncontrolled cell growth, which is cancer. Okay, so it says explain how the presence or absence, and that's all you really have to say. Let's see how they word it here. It says the response indicates that the lack of the appropriate cyclin-dependent kinase inhibitors, which are 18, 21, and 27, apparently from this data, might let cyclins and cyclin-dependent kinases continue to form active complexes that will result in almost continuous cell cycling, all right, with DNA replication and cell division. So we can look in, we can look in at this thing and say, okay, if the cell is continually dividing, all right, that means we must be missing the appropriate cyclin-dependent kinase inhibitors, which in this case appear to be 18, 21, and 27.